Thank you very much. Oh, thank you. So, hi everyone. In the next few minutes, I would like to talk to you about the uptake and clearance of EVs in the blood. I would start with a simple consideration. As you know, the total body mass is different in males and females, but approximately 45 to 40% is constituted by solid organs and 55 to 60% is constituted by fluids. Now, if we take this fluid compartment, 2/3 is intracellular and only 1/3 is represented by the extracellular fluid. However, 80% of the extracellular fluid is the interstitial fluid, and only 20% is represented by the blood plasma, which suggests to me that this interstitial fluid, which gives rise to the lymph, is a very important component, most probably, of the blood plasma EVs, since, as I mentioned, the interstitial fluid is drained by lymphatic vessels and lymph, and it's introduced to the systemic circulation. So indeed, we should remember that most probably tissue-derived vesicles are very important components of blood plasma EVs. In certain tissues, like in epithelial tissues, cells are in close vicinity with one or to one another. The vesicles, if they secreted, they are not supposed to travel too far because the neighboring cells can pick them up. But in other tissues, typically cells are at a distance from one another, and it really depends on the viscosity of the interstitial fluid, how efficiently and how far they can travel by passive movements. To exert paracrine modulatory functions. And in certain tissues, like in the different connective tissue types, it may happen that there's a dense extracellular matrix with protoglycans and collagen fibers, and vesicles are quite efficiently retained, and they have a hard way to move in this very viscous gel-like matrix. Now, we already do know that the extracellular vesicle membrane and extracellular matrix components have multiple different interactions with one another. We know that there are covalent interactions like tile-based interactions or Schiff-based or Michael adduct-based interactions, and also there are hydrogen bond and electrostatic interactions between the vesicular membrane and the matrix. How far the vesicles can travel in the matrix is also related to the deformability of vesicles, which is actually in association with the lipid composition of the vesicular membrane, and also it might be due to the aquaporin channels present in the vesicular membrane, which enable vesicles to adapt to different osmotic conditions. Furthermore, we have to assume that there must be reversibility in cross-linking of matrix components, which enable vesicles to traffic inside dense matrices. If there is an inflammation, as you all know, enzymes are released from cells, and these enzymes are efficiently degrading the extracellular matrix, leaving space for extracellular vesicles to be released from the degrading matrix in a synchronized manner. I would like to mention that just like we heard from Pia, there's a corona formation, and if there is an extracellular matrix degradation, the fragments of the degrading matrix components can be carried out on the membrane of the vesicles as part of their coronas. How do we know that indeed in these tissues, different tissues, in different matrices, we have vesicles? If you apply protease K digestion to commercially available matrices or real normal matrices, at the end you end up with vesicles, suggesting that these vesicles are there and they are retained within the matrix of these specimens. If the milieu is liquid, and there is not a high viscosity of the matrix, you can have a very efficient passive trafficking of vesicles if you insert an implantable little diffusion chamber into the peritoneal cavity of mice. And the top and the bottom of this chamber is covered by a porous membrane. Inside the chamber, you have GFP expressing live cells. The vesicles released by the cells can exit the diffusion chamber, and after 24 hours, you can appreciate that the vesicles are recognizable in all kinds of cells of the peritoneal cavity. So in fact, they can traffic there. But if you take a very solid tissue like myocardium, which is a rubber-like solid organ, even in this case, one can demonstrate, like we did recently, that using a double fluorescent reporter mouse model, cardiomyocyte-derived green fluorescent vesicles are detectable in the circulation. So even cardiomyocyte-derived, myocardium-derived vesicles can reach the blood plasma. So overall, if we are dealing with a tissue where cells are in close vicinity to each other, like in epithelial tissue, where cells are efficiently taking up secreted vesicles from one another, or they are cell-released vesicles which are formed in an extracellular matrix-rich environment, besides the local uptake by cells, we have to consider the interstitial fluid and lymphatic derived trafficking, which ultimately brings the vesicles into the systemic circulation. Beyond that, we know from zebrafish studies that there is an endothelial crossing by extracellular vesicles through the endothelium of capillaries from outside in. And these capillaries, again, bring the vesicles to the systemic circulation. And the circulation brings these vesicles through capillaries to different organs like liver, lungs, or spleen, where the vesicles have to go into the different direction. They have to exit the blood vessels. They have to move across endothelia to get out of the blood vessels and reach the cells in these tissues. The EV delivery routes and EV uptake mechanism, you all know, include clathrin-dependent endocytosis, macropinocytosis, lipid raft-mediated internalization, caveolin-mediated uptake. In the case of very large vesicles, even phagocytosis. And in a few cases, there seems to be evidence for membrane fusion as well. If the tissues release and contain vesicles, as I mentioned earlier, it's the lymphatic vessels that lead and drain the interstitial fluid and lymph into the regional filters, the regional lymph nodes. And from the lymph nodes, the efferent lymphatic vessels drain into larger lymphatic trunks and ultimately into the systemic circulation. I would like to also add that the endothelial lining is differentially permissive for the transition of extracellular vesicles. In most organs, we have a continuous endothelial internal lining of capillaries. However, as you know, we have fenestrated and sinusoidal or discontinuous capillaries in different organs. Like fenestrated capillaries are present in the intestines, kidney, and pancreas, whereas the discontinuous sinusoidal capillaries are present in the liver, spleen, and bone marrow. And these have gaps of 60 to 70 or 50 to 200 nanometers, which might permit the trafficking and transmission of the deformable small extracellular vesicles through the blood vessel wall. Accidentally, Pia showed the very same picture. I think it's very educational because, as you can see, both in acute and chronic inflammation, there are actually gaps between the endothelial cells, and everybody knows that in cancer we have a leaky endothelium, which shows a higher permeability for extracellular vesicles to enter or leave the circulation. A very old but very informative experiment from Eric Boylard's group from 2012 shows that if you compare control and arthritic mice and you inject them with different size polystyrene beads, you can very clearly appreciate that in the arthritic joints and arthritic inflamed limbs, you have an accumulation of 0.45 micrometer and even of 0.84 micrometer size polystyrene beads, very clearly showing an inflammation related permeability change in the blood vessels. Now, a big question is how vesicles are transported across the endothelium. This is a very complex question and it's under intense evaluation and investigation. What we do know that there is a mechanism of paracellular transportation. There might be some intraendothelial fenestrations in cells. And also, very importantly, we have to consider transcytosis, which means that vesicular transport from endocytosis leads the cargo across the endothelial cell and gives rise to the release of the cargo on the other opposite side of the endothelial cells. It is actually shown for lipoprotein particles like LDL. And given the fact that LDL and extracellular vesicles have been shown to associate with one another, actually bind each other, it is reasonable to consider that the lipoprotein EV complexes are jointly transported across the endothelium by transcytosis. In some way, you can consider lipoproteins as components of the EV molecular corona. So at the end of the day, the circulation via capillaries brings the extracellular vesicles to different locations where the vesicles leave the circulation and are taken up by cells of different organs. We don't exactly know the mechanism of exit of vesicles through the endothelium, but beautiful studies from the zebrafish world provided evidence that similarly to leukocytes, which exit the circulation by diapenesis, we see in certain regions rolling of EVs on the endothelial surface, suggesting there must be specific interactions between the vesicles that exit the circulation at a given spot and the local endothelial cell surface expressed molecules. The in vivo biodistribution analysis of small and large EVs showed the following results. In a recent publication, a summary, a collated result was published about the biodistribution of small EVs injected IV from 7 studies and large EV biodistribution from 3 studies. As you see, in the first hour, we have an accumulation of vesicles in the liver and in the lungs primarily. Between 2 and 12 hours, in the case of small EVs, the accumulation is most pronounced in the liver, and then with time at 24 hours, it's more concentrated in the lungs. There's a difference when we compare it with the large EV biodistribution. The accumulation seems to be slower. However, between 2 and 12 hours, there is a very pronounced accumulation in the liver, which stays even after 24 hours. Now, if we look at the ex vivo biodistribution analysis, that is when we look at removed organs, and this is a collated result from 36 published studies, we see that again, the liver is a primary organ where we have the accumulation of injected EVs as early on as in the first hour, and it seems to be present, the accumulation of EVs even after or above 24 hours in most of these experiments. Which are the cells in the liver that take up the vesicles? we found that even though hepatocytes constitute something like 60 to 70% of the liver total mass and cell number, we find a relatively smaller or lower contribution of EV uptake by the hepatocytes. It seems that the 21% liver sinusoid endothelial cells are the most active in the uptake of small EVs, and the professional phagocytic Kupffer cells, constituting 8.5% of the liver cells, are the best in uptaking medium or very large EVs. When it was looked at how long do injected vesicles stay in the circulation, this left-hand side figure shows you the pre-injection status. As you see, even after 2 minutes, only 30% of the injected vesicles stay in the circulation, and almost by 30 minutes, that is half an hour, a little proportion remains. 3.3 to 1.8% remains, while within 1 hour and above, hardly anything is left in the circulation. That tells you that there's a very rapid removal of the vesicles, injected vesicles, from the systemic circulation. The authors of this paper suggested a two-phase decay model in which there's a rapid distribution phase when the blood circulation distributes the IV-injected vesicles to the different organs. That's why they are absent in the circulation after a very short while. And there's a secondary elimination phase by different organs like the liver or kidney, which takes longer, between 34.6 to 184.5 minutes. Why are these organs, liver, kidney, spleen, and lungs, are the ones which show the highest role in EV accumulation and elimination? First of all, some of these organs, as I already mentioned before, have fenestrated or discontinuous endothelium, which enables the very quick and efficient exit of vesicles from the circulation to the tissues. These organs are known to be involved in filtration and clearance. They have particularly rich vasculature, and quite a few numbers of the mononuclear phagocytic system are present in these organs. Probably these are the explanations why they are the organs that play a pivotal role in EV removal. Interestingly, if we compare oral and systemic administration in mice, we see quite different patterns. After oral administration of small EVs, as you can see, instead of having a primary accumulation in the liver and the lungs only, we have quite a bit of accumulation of orally administered vesicles, also in the gastrointestinal system and in the lymph nodes that drain the gastrointestinal system. Everybody knows about the classical experiment of David Leiden's group, where the organotropism of tumor EVs have been shown to be related to EV surface integrins directing either lung or liver-directed organotropism of vesicles. And I would like to also, again, refer back to the EV corona. In a recent work, Samir El-Andalusi's group showed that if they engineered cells to release vesicles with albumin-binding protein, these vesicles accumulated a lot of albumin as an artificial corona around their surface. And these vesicles, these artificial corona-coated vesicles, had an extended circulation time and lymph node accumulation in mouse models. There are numerous EV imaging modalities, from fluorescent imaging through bioluminescent imaging, SPECT, PET, MRI, CT, and photoacoustic labeling. But recently, the multimodal, multi-resolution palm grat labeling has gained a lot of traction and attracted a lot of traction. That was published by the group of Charles Lie. In this system, as you can see in the schematic drawing, from cells and EVs and even non-EV particles up to in vivo whole body studies, you can use the labeling. And finally, I'd like to show two applications in the work from Ken Whitworth's lab in primates, complete pharmacokinetics and biodistribution analysis of extracellular vesicles was carried out using the PalmGret approach. And in another study, the PalmGret labeling and biodistribution approach was compared with the DIR lipid staining, and as you can see, there is tremendous difference. So it might be worth keeping in mind that the approach what we use for biodistribution analysis does make a big difference and could be a source of artifactual findings. I would like to quickly introduce to you my team members. And finally, I would like to advertise the EV biomolecular corona workshop, which is the next workshop of ICEV. It will take place next year in Italy, and everybody is welcome to join. Thank you for your attention.